I just want to say hello to everyone, and I would like to welcome all you here. And we are going to start the third day of iSCAN. Our first speaker is Nicolas Schuck from Max Planck Institute Berlin, and he's going to talk about the effect of age and L-DOPA on direction signals in human brain. The floor is yours, Nicolas.
All right, can you all see this?
Yes.
Okay, excellent. So hello, everyone. Thank you very much for inviting me to this really nice symposium. And today I'm going to tell you a little bit about our efforts to understand heading direction signals in the human brain using fMRI and pharmacological interventions. And what we were specifically interested in is to understand the effects of age and dopamine on these heading direction signals. So before I get started, I wanted to give you a little bit of a sort of background and also mention some of our earlier work that has led us to these studies that I'll talk about in more detail. So you all know that spatial navigation declines sharply with age. What you can see down here is data from 2.5 million people who signed up to download an online game. And what is depicted is an overall composite score that depicts their spatial navigation ability as displayed when playing this game. And you can easily appreciate that there is a pretty steep decline with age that starts also particularly early. So even if I look in my age bracket, I can appreciate that I'm probably vastly declined from where I was when I was around 20. And it's also true that it's not only the ability to remember specific locations, but also the way in which older adults navigate compared to younger adults is greatly different, as you can see from the plot on the right side, where you could see paths of a typical older adult and a younger adult who are trying to navigate first from A to B and then from B to C. And you can see that the older adults take a more tortuous path. So this overall decline in spatial navigation ability is made perhaps less surprising, but clearly also harder to understand when you take into account the complexity of the underlying neural systems that support this complex skill. And over the past days in the symposium, we've heard a lot about the different aspects of the hippocampal entorhinal system, where people have identified various spatial cells, including place cells, grid cells, but also head direction cells, and so forth. And we've also heard that even outside of the hippocampal entorhinal system, there are brain areas which make quite meaningful contributions to spatial navigation ability. Most prominently, perhaps, are the retrosplenial complex areas in parietal cortex and even the striatum and visual areas. So that of course, that makes it more, or begs the question, which of these many different components is impaired in age, or maybe all of them are, and how that specifically relates to the kinds of behavioral impairments we see. So now in human imaging, when people started to look at differences between older and younger adults when they do some sort of spatial navigation task, then what was typically found is a mix of over and under activations. You can see a really nice example of that here on the slide from a study from Moffat et al., where you can see that in this case, what they found was that in younger adults had a higher activation in the hippocampus compared to older adults, as well as in the retrosplenial complex here. But older adults had higher activation in the medial prefrontal cortex instead. So in our own work, we've also looked at this in the past, and I'll briefly show you a video of the kinds of tasks that we are using so you get a sense of that. And in these tasks, typically, participants are navigating using a joystick in a virtual environment that looks like this one. And they see some distal cues like these mountains that help them orient themselves. And they are typically asked to remember the locations of various objects. In this case, they are first exposed to the location of this pumpkin here, and at a later point in time, they will be asked where they have seen this pumpkin. So just like before, when you just look at overall activation differences between younger and older adults during different parts of the task, you will find brain area activation differences that are broadly consistent with what has been reported before. So in this case, we also found that younger adults have more activation in the hippocampus than older adults, and older adults instead have actually higher activation in quite a large number of different brain areas, including the striatum compared to younger adults. What we wanted to know then is to understand these brain area differences more precisely in two ways. The first one was we wanted to understand more precisely how younger and older adults differ in how they are doing the spatial navigation task, in particular which cues they rely on. And then we wanted to understand how activation in different brain areas relate to the differences in spatial strategy or the cues that they are relying on, and whether that relationship in and of itself might be changed with age. So what we did specifically in this study is that after an initial period in which participants navigated in this environment that I've just shown you, we did one of two things. Either we took a intramace cue, there was a traffic cone that was always standing in the same position during the training period, and we moved it to a different position. We didn't tell participant about it, it was just all of a sudden in a different place. And in the other condition, we just made the overall arena size either larger or smaller by extending or shrinking the boundary. Now, when you look at behavior, what becomes very clear is that younger adults are very sensitive to the change in the boundaries. And we can see that by looking at the change in the remembered locations before versus after that boundary change. So this is shown in this plot down here, where you can see the location before versus after the boundary change. And what younger adults do is that when the boundary is extended, they shift their memorized location slightly outwards. So they're keeping a roughly similar distance to the nearby boundary as before. And when the boundary shrunk, all the memorized locations kind of shift inwards in a radial fashion. Older adults, as you can see next to that, they react quite chaotically to that change, and there's no discernible pattern in how they are affected by this boundary change. So that difference between younger and older adults becomes more interesting when you look at the difference in the landmark condition, where older adults now show an ability to react to these kinds of changes in the environment. And namely, they systematically now change the memorized location along with the intramace cue. So if you shifted the intramace cue in a particular direction, they would show a tendency to shift their memorized locations also along with that cue. And that ability, in fact, is not different from what we see in younger adults. So they do that to the same degree that younger adults do that. So now the most interesting aspect then perhaps is to ask, how do these brain activation differences that we saw before relate to those differences in which kinds of cues participants are using in order to navigate? And what we asked specifically is whether the extent to which someone reacted to the boundary versus reacted to the landmark was related to brain activation during encoding the initial object location. And doing this analysis, what we see in younger adults is something that is quite consistent with previous literature in humans as well as in animals. Namely that the more hippocampal activation a younger adult had during encoding, the more that younger adult shifted its memorized location when we stretched or shrunk the boundary in a pretty systematic fashion, in a radial outwards kind of movement. And that's consistent with previous animal studies, amongst others, because there people have looked at how place cells shift their place fields when boundaries are changed. And they've also sort of seen similar stretching and shrinking effects. In older adults, strikingly, we found something quite similar. So we didn't find any relationships to the boundary changes, which is not surprising because they were almost random. But we found that the more older adults activated their hippocampus, the more they reacted to the landmark. So now, apparently, the hippocampus was related to processing a different aspect of their spatial navigation environment. So Following up on this, we wanted to ask about the other components that you need in order to navigate spatially in this task that I have shown you. And what we wanted to focus on specifically is our head direction, so our heading direction circuits. And as you all know, head direction cells have initially identified in the post-subiculum, but they have afterwards been found in the thalamus, retrospinal cortex. There's even some evidence that suggests that visual areas process heading direction related information, which relate to optic flow, that is perhaps also not very surprising given the strong influence that visual information has on these heading direction cells that we've heard about earlier during the symposium. And so these heading direction cells typically show a tuning towards a particular direction space, and they have been found in animals, and sort of recent work from Thomas Wolbe's lab has also derived nice neuromarkers in neuroimaging for identifying these heading direction signals. What we then wanted to know is, are these heading direction cells or signals impaired in the human brain as well? And that seems like a likely hypothesis, given that we have seen previous studies that were also already mentioned during the symposium that showed there's impaired grid coding in older adults, there is impaired path integration, and I think both of these indirectly suggest that heading direction might be impaired as well. But we also wanted to go beyond just asking whether it's impaired or not, but ask whether a specific phenomenon exists in older adults that has been observed previously in other contexts. And that phenomenon is called dedifferentiation, and it's perhaps best illustrated by kind of, again, starting with the animal literature, where it was observed that in older animals, cells that have a specific tuning function show a broader tuning width of that tuning function. So what you can see here are recordings from visual cells that show selectivity to visual orientation. And the bottom part is from a young monkey, and you can see that this cell has a sort of very clear orientation selectivity towards a particular orientation. And a typical cell in an older monkey has also selectivity, but a much broader tuning curve. In human neuroimaging, what has been observed is something that is related, but I guess not quite the same thing, namely that there is cross-category and cross-area confusability that increases with age. So that specifically meant that if you have one particular brain area from which you could, say, decode scenes, and you have another brain area from which you can decode very well which object is shown, then you would see that in older adults, the difference between what the scene area and the object area are encoding is diminished, in the sense of you can now all of a sudden maybe perhaps decode a little bit more about objects from the scene area and vice versa. And in essence, they become less specific to a specific information content. So what we did in order to ask this kind of questions about diminished tuning functions of heading direction cells, heading direction information in older adults, is that we went back to this original test that I showed you before, where we scanned younger and older adults, and they were walking continuously around in search and memory of these 5 different objects. And what we first did is that we took their continuous paths and we segmented these paths into discrete events. So there we basically looked for periods within which the participants were walking in a more or less stable direction within the same 60-degree bin for at least 1 second. And that meant that we excluded, for instance, time periods during which the heading direction was changing on a very fast time scale. And that's related to the sluggishness of the fMRI signal and the fact that we can't pick up easily, at least on sort of very fast changing signals. So with these events in hand, what we then did is that we wanted to try and train a classifier to predict which direction our participants heading in based on their brain data. And in order to not just ask whether the classifier works well or whether it works better or worse in older adults, we also looked at the confusion matrix of the classifier. That specifically meant that we looked at how often did the classifier predict, I believe this is direction 180 degrees, when in truth it was 300 degrees or 240 degrees or 120 degrees and so forth. So by doing that for all of the sort of different directions, you can derive something that is akin to a tuning function, namely based on the confusability of the classifier as a function of how different the angle was in which they actually walked relative to the direction that the classifier was sensitive to. Doing this is difficult in fMRI, again, for reasons that relate to the sluggishness of the fMRI signal. And in particular, this means we have very high noise autocorrelation over time. And in practice, that just means any signal that you will record in fMRI will be very similar to any other signal that you'll record a few seconds afterwards, just because there is high autocorrelation in the noise. So on top of that, it seemed very likely to us, and it's also empirically true, that participants are not changing their directions randomly, but rather it seems more likely that when they are heading in direction 60 degrees, then the next event would be in a neighboring direction, so 0 degrees or 120 degrees, rather than a completely random one, like opposite 240 or so forth. So with these two facts combined, it could be that we would see these tuning functions just from the combination of noise autocorrelations and kind of autocorrelations in the events as they happen. And to counter that, we simply divided the events into an odd set and an even set and trained separate classifiers, such that in essence, the classifiers were trained on events that had some meaningful amount of temporal separation, and therefore the temporal autocorrelation couldn't fully account for our findings. Now, when we just look at overall decoding accuracy, what we see is that decoding heading direction works best in early visual cortex. That's abbreviated as EVC over here, where we can decode quite well. It also works reasonably well in line with previous literature in the retrospinal complex. We found some evidence that we can also decode in the subiculum and in a mask that composed the entorhinal cortex and the hippocampus. There was significant against the permutation test, but it was kind of weak effects. And then for this first study, we wanted to be conservative and focus on the first two areas where we found stronger evidence. Now, the first thing that we did, as I mentioned, is that we looked at whether we would find any evidence that was non-trivial for these tuning functions. Non-trivial because we did this kind of odd even set test splitting, and we also did participant-wise model fitting, where we tried to distinguish whether the confusability is indeed a smooth function of angle or just a peak in the middle and then something that is described as sort of flat confusability. And that was true in both the visual cortex and the retrospinal cortex, as you can see with these two curves over here. But when we looked at the age difference, quite surprisingly, what we saw is that there was no difference between older and younger adults in these heading direction signals in the retrospinal cortex. And that is maybe best shown here by just seeing that the derived tuning functions have equal width in both age groups. In visual cortex, we find a clear difference between younger and older adults, where we can both decode better, but we also find specific evidence for a sort of flattened tuning function of heading direction. So now we wanted to know more about the amount of visual influence on these tuning functions or on these brain areas, the heading direction signals we found in these brain areas. And for that, Christoph Koch, who was heading the study, kind of came up with a clever trick, namely to look at periods in which participants were walking backwards. So because they were navigating with a joystick, that was quite easy for participants to do. And these periods are interesting because now participants are viewing one direction, but they are actually walking into the opposite direction. And you can take the classifiers that were trained on forward events and ask which direction is the classifier predicting now, the one that they're looking into or the one they are walking into. So by subtracting those, you can find a visual influence score. And the visual influence score, not surprisingly, is higher in visual cortex than in the vitreospinal cortex, as you would expect. But interestingly, we also saw that in younger adults, the visual cortex seemed to be a bit more sort of affected by the visual information than in older adults. And using a somewhat different approach that I can't explain here for time reasons, we also found some evidence that in fact, in turn, the retrosplenial cortex in older adults was a bit more affected by visual information than it was in younger adults. So this is kind of akin to what we've seen before, that in younger adults is a more or less clear-cut depiction of visual cortex being very dependent on visual information in coding heading direction, but retrosplenial cortex not so much, versus in older adults, this was a little bit distributed over both brain areas. So finally, we wanted to understand what effects dopamine has on these age differences in heading direction information. And we asked specifically about dopamine for two reasons. The first one is that sort of earlier theoretical proposals that had thought about why we see this dedifferentiation in aged brains have proposed that it might be due to a lack of neuromodulators that effectively change the gain of neurons and therefore cause sort of less clear activation profiles of these neurons. And the most prominent proposal comes from Shu-Chen Li. And on top of that, it's also a well-known fact that older adults do show a very marked decrease in dopamine availability with age. So what we did, therefore, is that we asked again participants to play a virtual navigation game very similar to the previous ones. This time they came in for 2 sessions. One session where we gave them a dose of L-DOPA and one session where we gave them a placebo. The order of the sessions was, of course, counterbalanced and everyone was blind. So the experimenters didn't know which one is which. And we have a somewhat larger sample size now of 30 younger adults, 40 younger adults and 37 older adults. So the first thing to note is that the L-DOPA manipulation in the dose that we gave it didn't have any overall effects on performance in terms of how well they could remember the locations of the different objects we had shown them. And when we looked at just decoding accuracy averaged over both sessions, then we found a picture that's quite similar to what we've seen before. Namely, we can decode reasonably well in the early visual cortex, where we also find big age differences. We can decode above chance in the retrosplenial cortex, and we find very weakish evidence that there might be something in the hippocampus ROI. Most interestingly, though, when we now looked at the effects of the L-DOPA manipulation, we do found that in the retrosplenial cortex and the hippocampus, L-DOPA indeed has a positive effect on decodability of heading direction information. So we can decode better when participants are in the L-DOPA session and not or almost not when they are in the placebo session. That was not true, interestingly, for the visual cortex. And now when we look at the age differences in this effect of L-DOPA, then we see that in the hippocampus, we see evidence that L-DOPA affected younger and older adults heading direction information encoding equivalently. We saw effects in both age groups. For the retrosplenial cortex, curiously, we only found evidence in younger adults, which is somewhat different from what we had predicted, because given the lower baseline availability of dopamine, studies typically report stronger effects of dopamine on older adults as compared to younger adults. Finally, we also, of course, checked whether the tuning functions are affected by age and by the dopamine intervention. And we saw the same age effects that we saw before in the previous study, but we didn't find any effect of the L-DOPA intervention on this metric. Okay, so with that, I'm coming to a close. And the summary is that I showed you some data that tells us that age affects heading direction decoding in early visual cortex, but not in the retrosplenial cortex. Dopamine affects heading direction signals in the retrosplenial cortex and the hippocampus, so in those areas where we do not see any age differences. And we also showed you some evidence that retrosplenial heading direction signals might be more visual in older adults than they are in younger adults. Finally, I showed you some evidence in the beginning about age-related changes in boundaries versus intramace cues. And the general conclusion there was somewhat similar in the sense that what the hippocampus did in older adults seemed to be somewhat different than what it did in younger adults. So with that, thank you. Worth noting again, sort of my PhD student, Christoph Koch, who's also here, who has been heading all of these studies, and our collaborator, Shu-Chen Li, who's really behind the dopamine study, our funding, and of course, my lab.
Thank you, Nicolas. Very nice and interesting work. We have one question from Matthew. Do you think that the fact participants couldn't have a vestibular input during the task, i.e. desktop VR task, would explain the reduced decoding accuracy on RSC, thalamus, or EC?
Yes, that's a very good question, and we can of course not exclude this possibility. So in fMRI, it's impossible as of now to expose participants to actual sort of vestibular information. So we don't really know, I would say. I think I would also even go as far and admit that it's possible that the mere fact that they didn't have any vestibular information at their hands might have changed how much responsive areas like the ERC are to visual information rather than vestibular information. But yeah, it's a good question.
Okay, any other question? Okay, I have one question. Probably you check this. Do you think that there is like a sensory input effect because the old people, some of them, they may have like eye problems? Is it effective on your results?
We screen all of our adults, of course, for eye problems. We correct them as much as we can in the studies, but of course there is noise, and I would doubt that if you measured it very, very carefully, that the visual acuity in older adults might be on par with those of younger adults. But that is a really interesting question. So I think follow-up studies could look at precisely how visually impaired are older adults, and how in turn does that affect how well their brain can encode visual heading direction, which often is derived from visual cues.
Okay, thank you. One question from Motoharu. I thought old people rely more on the borders for navigation, but what you showed was that old people cannot code borders.
Yes, so this probably refers to the results that Thomas Wolbers has shown earlier. And I think it's there's a difference between what he quantified and what we quantified. So he looked basically at whether participants have a biases towards lingering around the borders, if I remember correctly. And we looked at something quite different, namely how much do they update their memory representations if you really change these borders. And I think what we probably have to do is to tease apart what the precise reasons are behind spending more time on the borders. We also see that in our data. We also see that participants walk more in circles, almost like animals do, older participants in virtual environment. And it's not quite clear to me what that means in terms of encoding information that is related to the boundary relative to intramace cues. But there's definitely interesting studies that we should do to figure that out.
Okay. We also have one question from Jonathan Brown. Jonathan, go ahead.
I get to ask mine in live, in real time. So I've just, perhaps I missed this. What was your definition of young and old? And is there do you see intermediate effects in the sort of, in the middle age?
Yeah, I didn't mention it. It's a good point. Youngs are typically between 20 and 35. Old is 60 upwards. We haven't looked at intermediate age groups in this sample because of the constraints of fMRI, but we have done behavioral work. Shu Chen Lee has done behavioral work where she looked at all the different age brackets only behaviorally. And there we see indeed that there is a pretty continuous change over the lifespan that goes from being relatively intramace cue dependent in kids to being more boundary dependent in middle-aged adults, and then it kind of goes down again back in old age.
Okay, maybe the last question from Caswell.
Hi, Nico. Thanks for an awesome talk. I just wondered if you could speculate on what sort of the neural basis of your RSC ability to decode direction of travel is, because you started talking about head direction cells at the beginning, but of course head direction cells would tell you the direction you're looking in. So just curious what you think that might correspond to.
Can you, hey Caswell, can you repeat this? I think I didn't really caught the gist of the question.
Yeah, so you showed you had this really nice control where you get the participants to walk backwards, and in retrosplenial, your decoder is able to decode the direction of travel, not the direction of facing, or rather there's a significant difference between those two things. I'm just wondering what you think that might correspond to on a neural basis, because you started at the beginning talking about head direction cells, but of course those would tell you the direction you're facing, whereas this is more like the direction of travel.
Yeah. So it wasn't that we could decode the direction in which they were actually walking in terms of the backward decoding. It was just that direction was less decreased as compared to what we see in visual cortex. On visual cortex, it's sort of a quite sub-baseline effect because visual cortex is clearly predicting the direction in which they are viewing. Retrosplenial cortex seemed to be a little bit more confused in the sense of predicting a bit about what they're viewing and a slight reduction in the sort of walking direction, but it wasn't affected as strongly as visual cortex was by the conflicting information pieces.
I see. Okay, thanks.
Sure. Thomas, I can answer. We can chat offline if you want about your question. Thank you.